That's what I'm about to get to. I was gonna pull up our PowerPoint.
Where's the PowerPoint?
In my Google Drive.
Oh,
You can't pull it up?
yeah, you can. Yeah. You want to... Not for you.
Thank you.
So many emails.
Oh, can you save it? Save it on my own. File. Download? No. Wait, download as. Oops. Okay, look like it's already done.
You want us to change our name?
No. Okay. Yeah. Actually, that should be your name. Anyway. Oh, you're here. You said president. Yeah, that's it.
Did I do something?
Oh.
This is not my fault.
Yeah, okay.
That'll be the best. Okay, good afternoon, class. My name is Jamia Robinson.
I'm Mary Carmel.
And we will be presenting the paper that I'm sure everyone has read, Contrasting Evolutionary Patterns of Spore Coat Proteins in Two Bacillus Species. Groups are linked to a different cellular structure.
Okay, so one of the characteristics of the genus Bacillus is their ability to produce spores. And so in all the spores, they all have coats. Some of the coats are thin, some are thick, and they're the protein shells that encapsulate and protect the spore itself. And yeah, like different species have different coats. So in this project, we talked about the Bacillus subtilis and the Bacillus cereus. So in the subtilis, it's the primary model for the gram stain positive, and it's the model for bacterial development. And this spore coat has 3 layers. And then for the cereus, it's the foodborne pathogen, and the coat is really thin as compared to the subtilis, and it also has an additional structure known as the exosporium.
And the 3 layers are consistent of like a thin layer, and the middle section is like a lightly stained area, and the last layer is a darkly stained area. I tried to pull up a photo that kind of showed it, but it's kind of hard to see. And the exosporium is just an additional, it's very, very thin. It's the thinnest layer that either of the spores would have, and it's just the additional structure outside of the spore.
Okay, so for the methods, the first sequence they use 5 group of genomes, the Bacillus subtilis. Okay, these names are really long. And then, okay, so they analyzed each group, and then they did an inference of species reference tree and alternative topologies. And for that, they used the 16S rRNA approach and the multi-locus approach. And for the multi-locus approach, they chose a sequence concatenation-based approach, which is a list of the 34 essential genes. And for the last method, they did a statistical analysis and data visualization, and they did a sequence alignment done by the Clustal W. And then the drawings that they gathered, they either did them manually or they performed them with the mega and dendroscope method.
They also use this ortho. What is it called? Ortho. I don't know how to say this word, but however, it's ortholog. It's the ortholog. It's the identification of the ortholog, and they use this frame to go through each parts and use the protein blast. And here is where the coats are. Yeah, where the ortholog's identification of the coats occur here. Wait. Back to the PowerPoint. And they also did like phylogenetic profiling of the spore coats, where they used this big old little thingy. What did you call it? Because I don't know. However, it's kind of complicated. Bear with me right quick. This structure here, where they divided each of the clades into the two major clades, which is the subtilis and the serous, and the out group is just another little group of the Bacillus genus. And this just shows the grouping of the phylogenetic. Yeah. This is crazy. Can I just do this one and then go back to the PowerPoint?
That's not your PowerPoint.
Okay. So they used the phylogenetic-based approach to identify the orthologs and the distribution of the coat proteins to identify the evolution of the spore coats. It's complicated to show without the graphs. So, however, the hypothesis of this experiment was just to see if there are any differences in spore structures, then they can influence the spore coat protein divergence in evolution. The results that we saw in the paper showed that the evolutionary differences in the spore coat proteins can reflect their locations in spore coat layers and differences in the spore structures across the species. However, the phylogenetic profiles that they used in the graph, if you have the paper, is on page 3. The coats were divided into two sections, which was the conserved and the labile profiles. And they're divided by rows and columns. If you see the rows, they're showing the inner coats and the outer coats, and the columns are just showing the I actually need the PowerPoint. Okay. Yeah. So, like I said, they were showing the inner coats and the outer coats on the columns and the rows, and they found out that the inner coats are more conserved than the outer coats, which is on the Bacillus cereus, because the spore coat is interacting more with the environment rather than the 3-layered Bacillus subtilis. And it also has a higher non-synonymous and synonymous rate ratio in the B subtilis, which I was saying before. That's because it has the 3 layers, and it inside the inner layer is not as active with the environment. So that's why the B series is less synonymous, non-synonymous. The ratio is lower. Okay, and the co-proteins were partitioned, like I said earlier, this was just the last little part that was saying the conclusion was just the evolutionary conserved and labile profiles. And in figure 3, basically, I'll show it to you in a minute, but basically the inner coat is more conserved than the outer coat. And and they did a table that showed it shows the outer layer and the inner layer versus in the same species, which was the Bacillus subtilis. In this table here. And as you can see, in the conserved area, the inner coat has more non-synonymous, a higher non-synonymous synonymous rate ratio than the outer coat. I don't know. Oh, the conclusion.
Okay, so basically to summarize what Jamia just said, so the different co-proteins have various selective pressures, which she was saying like with the 3 different layers in the subtilis and then the thin layer in the serous. And so the absence of the exosporium in the B subtilis allow for a relaxed negative selection pressure on its outer coat.
And that concludes our presentation.
Okay.
I wish we had something to show like together.
Okay. Obviously, a lot of effort. Okay. Let me run over some of the. I actually have my own presentation. I thought it would be boring if I. Oh, here's my own presentation. So I actually used this in a job talk one time. So those are the major species I'm studying here. Oh, I see. Okay. So I started this project when the capital anthrax attack around that time. So you can see how long it takes for me to actually get published eventually.
Sorry. That's hers too.
So it turns out that this species have a lot of important, or this group has a lot of important species, including Sadolis. That's in fact, we use that in the lab for grandstand. So this is the gram-positive model organism. Anthrax agent series, that's actually most of the bread or food when you throw out, that's what grow on them. That's a series. Syringim is actually is important in agriculture because it actually kills some insects. This bacteria kills some insects. Okay, so that's the overall design of the study. I actually did a lot of work. Only a fraction of the work I eventually published. Some of the figures also changed a lot. I tried 3 journals eventually. So it actually went through about 4 or 5 rounds of revision. The funny thing is about the one journal said you should do this, and so I changed it. The next journal, next review, you should do the previous one. So all I did, I just switched the different version. Eventually, one version got it through. Kind of really funny in the end. But okay, so but the main work is just this. First, I identified autolog and see the pattern, and then compare the evolution pattern in the inner code and outer code, and then do more advanced study of them. So Oh, that's the bacillus. Bacillus is actually everywhere. In fact, it, on the dirt on the bench, it's probably also on your skin. It's everywhere. So sorry. The main place where you found the bacillus is in the soil. It's in the dirt. So okay, so there's actually a lot of species in the genus, but in the lab we can only study a handful of them, and they can be separated into two groups. That doesn't look right. Something didn't come out right. Sorry, but there seems to be some typo over there. I don't know when did I get into it. Sorry. Subtlest group and a serious group. This one doesn't. I'm going to show the next. Oh, sorry. Let me. What I see on my screen and what the display are not the same. So there's probably some adjustment I need to do. Yeah, this is what I want to show. For some reason, the previous one only show a fragment of this. I don't know why. But okay, so those are actually the species I studied in the eventual public work. So 1, 2, 3, 4, 5 species in the Satellite group. 1, 2, 3, 4, 4 species in the Series group. The funny thing is about when you talk about species, human and mouse and dog, it's very obvious we are separate species. In bacteria, it's actually not that clear, even for E. coli. Can you tell E. coli is a species? Actually, isn't that clear because in bacteria, only about 70% of the gene are conserved. The rest of the genome is fluid. You cannot imagine that for human, right? But for bacteria, that's actually the case. So at a certain point, you will say, well, we share 30% of gene. We are the same species. What about 75? Then 65, it becomes grayish in those areas. So, but at least so I'm trying to avoid those controversies, only look at the at least people agree upon. That's what we are focusing on. Okay, so it turns out that most of those are what you can call good cops or nice guys because they don't cause some major disease or doesn't cause other problem. But this group, this cause food problem, this cause anthrax, this cause some disease to some insects, and that one I'm not sure. But in general, those are having some problem. And those basically are outer group. The outer group are only used when we try to infer the root, the ancestral root of the different species. Without outer group, we cannot tell which one come first during evolution. That's the only purpose for this study. We use those two as outer group. Okay, that's the code structure. So I think your two classmates show there are two major layers of the code, and then the extra thin layer called exosporin on the outer casing. So it turns out this spore is resistant to heat, chemical, pH, radiation. In fact, even when NASA, one of the NASA founder, when they sterilize everything, there's still some bacillus spore found there. That's probably called Bacillus odyssey. They actually named after that the instrument because even after they do intense radiation, they still found some bacillus spore there. So they named after that instrument. Oh yeah, I actually have a slide on this. So I didn't put out. It's actually, if you Google on Wikipedia, there is a page to explain this. So that's actually one of the main challenges you want to find life on Mars. You don't want to bring life to Mars. So then So there's a lot of morphological difference in bacillus, but in general, those are the case. They are outer coat and inner coat for Saturdays. There's a lot of proteins, some in outer coat, some in inner coat. And there are actually some in between, some in both. Okay. Well, this is just some philosophical argument, I guess. Why I have to do this? If you want to do something, you have to explain to people. But I guess you all agree this is something important I should do. So, well, I already done it. So, so this is the actual work I did. So I think when I started, I actually found hundreds of genomes. And for me, it's actually not a big deal. And if I analyze 10 genomes and analyze 100 genomes, it's actually the same amount of work. All I do is just run some software at it. Let's say there's no one going to sell me that software. I wrote every line of code I need to do. So all this, I'll probably write 1,000 of lines of code for this work. But all those genomes going to belong to the different species group. So all I did, first found out 16S. In fact, Dr. Wenji Li from Morehouse also talked about 16S. The 16S is one of the most conserved machinery in cell. It's the main part of translation machinery, ribosomal. So it's actually highly conserved. Almost every cell has one. Well, actually, every cell has one. Otherwise, it just won't survive. And we found that, and then found out the essential gene. So there are about several hundred essential genes in bacteria. So those are the housekeeping genes. So also found out ortholog. Ortholog means the same gene in different species. That's the definition of ortholog. If it's the several copies of the same gene in the same species, that's paralog. Paralog. Yeah. So that means one gene duplicated in a species become multiple. Those multiple copies are called paralogs. So then infer the species tree using a concatenated essential gene. That's because 16S is so conserved, it doesn't have too much difference among them, so it cannot generate a resolved tree. In biology, a phylogeny always is bifurcated. For some reason, we cannot do trafficating. We don't believe trafficating are more than 2. And so it's always bifurcating. So we generate species tree and then have the list of the ortholog code gene. Then you basically, that's the work. We'll first do profiling and then compare the evolution rate. Profiling, after profiling, I can see that's probably the table 1 or table 2, the Fisher's exact contingency table to compare the inner code and outer code, which one has more ortholog. That's the based on this result. And calculate the evolution rate. That's the pairwise comparison in a heat map. And then this is the last figure using likelihood method. One of the last figure in the result. I actually tried to look for something called adaptive change, but that didn't get published. But I actually did some work on that. Let's see, those are basically the main finding. Okay. All right. Well, in general, if you want to infer how species is related, you can use a ribosomal RNA or something called a multi-locus approach. Basically, you find a lot of genes, conserved genes, concatenate them, see what's the most consensus topology among them. That's also that's generally is not a problem in higher organisms like mammalian or even fruit fly worms. Those are all well-resolved species. Most of the genes you took, they'll be very easy to do. But in bacteria, it's very difficult. Okay, so this is the 16S RNA tree, and that's the multi-locus species tree. So those are the out group. this is the shuttleless clade. That's the series clade. So those are the good carp. This is the bad carp. That's the outgroup. So, ortholog genes in different species share the same gene in the ancestral species. That's the definition of ortholog. So by definition, the ortholog should play similar functions. Okay, that's how I identify the ortholog. We can ignore that for the time being. So basically, there's a lot of technical, you can call that informatics, or you can call that molecular evolution. It depends on how people view it. Anyhow, but the point is, no one gave me a software. I wrote every line of the code here to do this kind of work. And of course, I also use a lot of open source code. So people provide other code for me to do this. And so this is a software someone else provided. I run a BLAST, of course, is NCBI tool. I run the BLAST on it. And neighbor join is a well-established algorithm, so there's a lot of way to do that. I also tried a lot of other software on this one. Yeah. So, okay. Why about the phylogeny? That's because phylogeny is really the most, well, at least most consistent way to do genome annotation. In fact, during the process, I also found some mistake in the database. In NCBI or other database, there's, once I have the phylogeny tree, I compare with the annotation, I see mistake in those annotations. Okay, so that's the overall pattern. Blue means ortholog, red means none. I guess you don't see clear. I don't see clear either on that figure, so it doesn't come out well. This one should, well, the most conserved one should be Bacillus subtilis itself, because I studied it. So this line must be Bacillus subtilis. And those are related to Bacillus subtilis. So this, on the right 4 panels, those are the subtilis group. On the left side, that's the series group. So even though you don't see clear, but there's some reason, there's some logic you can guess which one is which. So, and on the top you don't see clear, but there's actually a tree. Maybe on the paper you can see. That tree, basically using this profile, I can generate a tree. Basically, is a species tree. So those patterns are actually consistent with the species tree. Those are the conserved protein. Those are labile protein. And then, since I have the conserved and labile, I can then that's for comparison. Those are the essential genes. Those are the genes when it's missing, supposedly will kill the cell. That said, still there are a small fraction of them still missing in some of the species. So that actually could be a bioinformatics problem, because when you detect a protein, you have a threshold. That threshold is basically arbitrarily cut off. So although it's not detectable, maybe it is there, but I didn't detect it, basically. Yeah. Okay, so, and then that's one of the table. That's this table. Yeah. The idea is to compare the inner code and outer code. Do I see they are as conserved to the same degree during evolution? And so those are the ortholog hits, inner code and outer code. And this is basically 2x2 table test. You will see this a lot in social study, in comparison study. I mean, or survey data. You actually see a lot of this 2x2 table comparison, Fisher's exact test. If you Google, you will see the reason it's called Fisher's exact test because the Ronna Fisher talked to a friend. That friend claimed, and she can taste 2 kinds of tea. If you add milk first, or you add the tea first and then milk later, these 2 ways, if you mix the tea in 2 different ways, you can taste the differences. And so Ronna Fisher, well, say, how do we prove you can do this? And so she let that person try and compare the result. And it turns out the first time ever this method is applied, it's a significant result. So she proved that lady can do what she claims she can do. So it's kind of funny how this method ever applied. Yeah. Okay, but it's now called Fisher exact test. So basically, the idea basically say, well, we have the inner code and outer code. Which one is more conserved? And it turns out the difference is quite significant. The inner code is more conserved than outer code. And significant means it's not by chance. That unlikely. If it's happen by chance, this is 2 or 2.5 out of 100. Right? But this is so it's unlikely from that perspective. Okay, that's just more analysis. We can ignore that. So the main difference between subtlest group and the serious group is really the difference in exosporing. In subtlest, this exosporing is actually very, very thin, almost doesn't exist. It's actually called a different thing. But in anthracis, the exosporing is very conspicuous structure. But then its coat structure becomes much thinner. So the two features are reversed in these two major groups. So the argument basically, well, we see this, we also see the structural difference. I mean, it's correlation, but we didn't prove it. We basically see the difference and see also it's correlated with two structural change. Now, can we do more than just compare the autolog hits? So, well, so then we apply more molecular evolution technique to see the selection pressure. Here's a key concept you really need to remember. So this is not just molecular evolution. This is a general genetic concept. So if there's a mutation, if there's a change in the DNA sequence in the coding region, it may or may not cause change in the protein sequence. Right? And now, the consequence of this, if it causes a protein sequence change, that means evolution can act upon a protein change. If the DNA change leads to a protein change, it's more likely to affect the function of the protein. If it's a bad mutation, and then evolution will suppress it. But if it's a change, only affect the DNA level, not the protein level. The protein function more or less is still there. So it's hard for evolution to act upon that mutation. More or less, some people will argue that's called a neutral mutation. So those will be, or silent mutation. That will be the synonymous substitution. Why it's called substitution? Because, I mean, it's easy to say, well, this is a mutation, but in reality, if you compare the nucleotide genome difference from individual to individual, there's actually no wild type, unless someone says this is the reference people. I mean, then everyone else is a mutant. So there's no such thing of a wild type when you study population. Everyone is a mutant by definition, or everyone is a wild type. It depends on how you view it, right? So instead of commutation, we call it substitution, right? So all those substituting they can be either synonymous, means the amino acid are the same, or non-synonymous, means the amino acid will change. So that's basically it. So I guess substitution is a political correct word for DNA. Sorry, that's a bad joke. All right. But how do we tell selection? If something is selected, that usually means there's a purpose there. That's also a bad word in biology. You don't say purpose. Sorry. Actually recording myself. So it turns out the way you want to tell is if you want to tell something is good during evolution, you want to say it's adaptive or selective. But how can we tell? You have to compare this non-synonymous rate versus the synonymous rate. Because the synonymous rate is not something selection going to mainly focusing on. So that's your background rate. If something happens very high above that background, that means something has been selected. So we can this is our background rate. That's our observation. You want to see this rate very pretty high. If they are the same, that's be something we call neutral. And if it's small, that means negative. If it's above, this is a positive. We want to look for the positive selection, but that happens very, very rare. That is because most of the things in our body are useful or functional. So, we want to keep them there. If something happens there, most likely it's going to bad for us. So during evolution, this happens probably 99% of the time. Most of the genes are under negative selection. But every now and then, there is a very rare change actually gives some advantage. For example, like lactase persistence. Like people like me, in fact, I can guarantee a third of you also are lactose intolerant because we're not supposed to drink milk during adult time, but somehow during those dairy product domestication event, some people gain a mutation in their non-coding region, so they can express lactase in their adult, and so in a I don't know farming incident, they can drink the milk, and so that certainly give an advantage of that. So in that case, that's a selective advantage. Well, I actually complicated the thing already. We are talking about coding sequence evolution. The lactose persistence actually is a non-coding sequence example. Sorry. But in any case, the philosophy is still the same. Okay, so positive selection requires some new sequences. A negative selection actually preserves the existing sequences. That's the main thing. But most of the new change, most of them are bad. So that's why we don't see it a lot. But once we see it, that often means something very important. For example, at one time, people say, "Oh, we found a language gene." That's because we compare ourselves with chimpanzee. There's a DNA change in our genome linked with the speech feature of us, but not in the chimpanzee and other primates. So actually, if we found that, actually tells us something of it. Right? So, okay, this is my best effort so far to explain why we can do this. So if you think about a DNA mutation, basically, you think a DNA mutation only have 4 chances, right? A, T, C, G. And it's basically almost like a 4-sided dice. You drop it, it can be landed on either side of the wall. How does selection influence it? Selection itself never causes mutation change in DNA thing, but it actually increases the frequency. So it's kind of a counterintuitive. How can selection influence this random process? That's how this goes. So you think about the random mutation, basically randomly drop here. If I pull this drop to the other side of the wall, it's going to increase the chance of it landed on the other side, right? So although selection itself doesn't cause this mutation process, it actually can influence it. And once this influence somehow is called selection or adaptive change. Okay, and the same thing is except for nucleotide, that's the 4-sided dice. We drop it during evolution. If they cause, say, 50% cause synonymous change, another 50% cause non-synonymous change, that's actually not exactly 50-50 based on the codon biases. But if this one is, they are more synonymous, that's positive selection. If it's less, that's a negative selection. There's actually a very complicated algorithm to compute this process. It's not 50-50. It actually depends on the frequency of the codons. Every species actually have different frequency. Okay, so, and this omega is called selection pressure. This omega is probably one of the most important parameters to study population genetics or molecular evolution. In fact, one of the many software, when they want to analyze human genome, they will compute this omega. Okay. So what I did is compare the omega in the spore coded gene versus non-coded gene. And actually, that's what I first did. After I did it, and then the reviewer actually said, it's obviously the non-code gene will contain essential gene. The essential gene is going to be slower than code gene. So you haven't proved anything. You just proved the essential gene is more conserved than code gene. I thought, well, that's a good point. So I have to take the essential gene out from this comparison. So I remove the essential gene, compare the code gene with those non-code, non-essential gene. That's so I submitted, and then the reviewer said, no, this doesn't cut it. So I have to remove the essential gene from the whole analysis and redo the analysis again. I'm going to. This actually is the histogram of the omega. So this sharp one must be coded gene. Oh no, the sharp one is non-coded gene. The red one is the coded gene. The well-known is the essential gene. In any case, the omega of the code is to the right, means it's bigger than the essential gene and non-code gene. But I need to show this is statistically significant. So I just apply the t-test. And I then do this for every species, do a pairwise for every species. So I have all these species here. And of course, if you compare the species to itself, that doesn't make sense. So it doesn't compare to itself. That's why there's a blank spot there. And then I do this for all of them. This is a one-sided test. If the code gene shifting to the right, that means it's faster. If code gene shift to the left, it means it's slower. So I basically do a pseudo-color plot based on the p-value. So the greenish means it's slower. The code gene is slower compared to the rest of them. The red gene means the code gene is faster than the rest of it. And I'm actually very proud of this plot. I actually look at the previous publication. I don't see any similar study of this. But when I say this, the reviewer doesn't agree. But he doesn't provide anything else to say this is the first time. But I just ignore that. Well, I did this. Whether it is first time or not, but it is there. So, okay. So, but you actually clearly see in this series group, all the color are quite different from the rest of it. So that's this actually in my earlier version of plot. In the publication, you can actually put a species tree on the left. That is actually suggested by the one reviewer. So it's actually a very good suggestion. So I generated the figure when I did the first version, but if you look at the publication, there's a species tree next to it. That's added during the revision process. Okay, so far so good. I mean, I show this it's quite dramatic pattern compared to the rest, and all this actually didn't take much time. I spent this look like complex figure. It actually only take me a summer. I spent the summer at Washington, finished this figure, and this figure was done basically at a Barnes Noble coffee shop most of the time. But then the rest of then I try to do on this. This has taken me a very long time to finish, mainly because the result is not as clear cut as the previous one. So the study itself is quite. I mean, run a software compute is actually quite fast. It's basically mainly because the result itself take a long time to write out something meaningful. Okay. The main idea is those are the evolution. This is the main tree for this species. I want to compare the selection pressure, the omega, in different branches. If you look at the tree, the phylogenetic tree is basically like tree grow the other way around. In a tree, if you want to see one branch different from the other branch, what I want to compare is that selection pressure omega. So, and then how do I compare omega? So I have to generate a different hypothesis. So the null hypothesis, all the branches had the same omega. That's the H null. Every branch had the same omega. So that outer group, omega null. Omega S is for Shuttlesworth group. Omega C for the series group. They are all the same. That's the null hypothesis. So, and then the alternate hypothesis is the series group is different from the rest of it. Another alternative is the Shuttlesworth group is different from the rest of it. And there's a more complicated hypothesis called every omega is different. So this model is something called a hierarchical model or nested model. So using this model, we can apply something called likelihood approach using probability. So we can calculate the probability of all these model, all these scenarios can happen, then compare the likelihood and see which one is the most likely event. It turns out so this is a computing intensive approach. So I apply this to all the, basically I apply this to the genes in the entire genome. There will be 3,000 or 4,000 genes. I compared all of them. Oh, I didn't. Okay, I'm going to switch to the. I printed one of the last figure I didn't. So, and then I compare the omega in the Shuttlesworth branch versus the series branch. And the diagonal means they are all the same. If on the other side, that means greater than 1. On this side means less than 1. And the p-value in the paper say this is 0.07. Well, it's not significant, but what I argue is, well, it's not significant, but it's certainly not very high either. Besides, this is small sample size, because the sample, to do this, I have to have the genes in all the branches, and so that cut down the sample size. So what I argue is this is small sample size. It's not significant, but it's consistent with the previous observation, and the reviewer didn't argue against this, so finally this is published. So, oh gee. Okay, so I will just skip over the rest of it. Actually, also did some structural study too. So actually did find a few highly selective residue, but there's just a few of them. It's not allowed to publish. Yes.
Which one was the highest likelihood? Which hypothesis?
It depends on which gene. So for every gene family, I compare the 4 models. So, right. So for every gene, I compare the 4 models. So H null, H1, and H2. And so I started how many genes I have? About this is only about 1,200 genes, and then among those, H1 are likely. H1 is better than H null. And then about 80 genes, H2 is better than H1. So altogether, there are about 200 of them actually can pass the test. So, and then, so the results, in this case, it's indeed quite confusing. And I also did the Venn diagram. The trouble is there are too many scenarios to describe this. So I mean that's now, and when things are different, they can differ in many different ways. So that's this graph basically describes all those scenarios. But how do we interpret it become really tricky. But finally, the reviewer said, I only want to see this diagram. If I see this, then it makes me actually that makes sense. So this I submitted A and B, and reviewer asked for C. Actually, it makes sense that from the way he write, I think he's British, but that's my guess. At least had a British background. So that's the result. the final conclusion is basically this. Let me see. So in the series group, we have the simple code, but has the axial sporing. In the subtlest group, we do not have the axial sporing, but have the inner code and outer code. So the argument is in the subtlest group, since the axial sporing is not there, so the outer code has more interaction with the environment and cause more labile change and faster evolution. So yeah, so that's this is actually probably my it's not my first publication at Spelman. But it's one of the most proudest publication at the Spelman. So I think I thanked let me see. You can see my acknowledgement. Yeah, I should also thank the provost office because they paid for the publication, but they paid after the paper already accepted. So I thank the Aspire program, thank the HHMI program, thank the RIMI office. But in retrospect, I should, well, it's Spelman College and name is there. So the provost actually gave me a check to pay for the publication. Okay, let me see. So, yeah. In my, I started, when I teach microbiology in 2010, I actually let the student discuss my first version of the paper. So now it's published, I guess, a different perspective. So, okay, so the version I give it to you, it's probably my third version. In my you can actually see the reviewer response all 2012. I actually tried a few journals, and so, one time is quite stupid. Apparently, one of my co-authors has one of the reviewers seems to be an enemy of my co-author and seems to be rejected out of a very stupid reasons. So I realized that that's the problem. I just switched to a different journal. So what time is it? Okay, I think I get most of the things done. So obviously there's a lot of technique to it, but at least I want you to understand some key concepts as covered in the multiple choice questions. So what do you think are most important concepts you want to cover? Ortholog, synonymous, non-synonymous changes, omega, how selection can be identified using omega, selection pressure, outer group, what are other things? Actually, p-value. P-value, how do we interpret the p-value? That's also it. But since many of you are taking MCAT or other tests, that's actually part of the required skill. Yes?
When calculating selection pressure, what is the derivative of N and derivative of S?
Oh, that's not derivative, that's the distance.
Okay.
D for distance of non-synonymous. Yeah, that's D and DS. It's not derivative. It's the distance of non-synonymous and distance of synonymous. Some people call it Ka or Ks. It depends on that's because there are many groups study this method, develop their own method, so it end up there are several conventions of this method. Okay, let me see what other things I want to do. So Monday we'll do a phylogeny computer lab. So I email you a link. If you can install that software beforehand, that would be really great. So we'll use a software called Mega for this. And if you just Google Mega, actually it doesn't give you. You have to add Mega evolution or phylogeny to find it. So Another thing I want to show is the striking colony. I think I finally. So Tamara helped me generate a video for this. Hopefully it's more informative. Let me see. Yeah, so here, striking for a single colony on plate. Flame the loop. Make sure that the tip of the loop is burnt red, and we should use the top part of the flame. Okay. And before we do that, we want to make sure the loop is actually cool. So I want to touch the side of the plate, make sure it's cool. Yeah. And then I take one single colony, let's say this one. That's actually a lot of cells. So this will be my first strip. So I'm going to, this is going to obvious, but I still need to remember it. So that's my first strip. That's actually a lot of cells. And then I'm going to burn this. So if you look closely, you can see that's my first streak. So I'm now doing the second streak. So this time I want to save some procedure. So I'm going to remember the side of the loop I'm going to use. So use one side to my second streak. So basically the loop, when I streak, the loop is vertical. I use and how should I flip? Wow. So I put the loop like this way, and then I flip it that way. So that's at least do it faster if you do a lot. So I streak, and then I flip to another streak. Yeah. One, two. That's actually a lot. And then I flip the loop. The other side of the loop has not touched any cell yet, so I'm using that one to my third streak. So I then flame the loop again, since I used both sides. Make sure it's red. And then you want to look at the mark, the streak of the first, my third streak, and then cool the loop again. Remember the side, this is my fourth streak. And then flip the side. Well, since this is going to be my last streak, I'm going to burn it and make sure it doesn't have some previous cells. So this time I'm going to try to spread all over the plate. So I will cool the loop first, make sure it doesn't burn the cell, and then going to try to this is my last streak. I do not want to touch the previous streak because the previous streak contains more cells. So okay, all right, and then burn the loop and sterilize it again. So if you can see first, second, third, fourth, and then fifth all over the plate. Okay, so I'm going to show how to done. Okay, that's I should put that also on Moodle, but yeah. So okay, so Monday we'll do a phylogeny lab, and then you have a first attempt of the practical striking single colony. So I can give you if you can. In the first 3 attempts, you can get A, you will get A. So after the third attempt, after 3 times, if you still want to do it, you can still get A minus, I guess. So
Okay, so you said, is this the practical?
for practical stricken colony, you have three strikes. If you get an A in that 3 strike, you get an A. After that, you still want to do it, you can still get A minus, but I will give you a passing grade since you always have tried after three times, right? I will give you a passing grade, but if you still want to try for a good, better than C, you can still get A minus after 3 trials.
So that's the only technique we're being tested for this.
Right, yeah, for this practical.
When is that?
We start on Monday, so yeah, Monday you have your first strike, and next, well, you can do it all the way till, right, yeah, so yeah, you don't have to do it in the lab time. You can just stop my lab and you want to work on it, right? So since this doesn't take time, you can see I only spend 3 minutes, and with explanation I took 3 minutes. So if you just want to do it, you can start my lab. In fact, Tamara made almost 30 or 40 plates. That's quite a lot. But you don't want to do it all at the same time because you don't know how it works, right?
Right. Okay.
Right, so yeah. Yes. Questions? Okay, let me see how many people are here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Okay, that's a lot of people. So this is a good time for you to do course evaluation, I guess. So I need a volunteer to do this.